Good morning, good afternoon, good evening, everyone. You are welcome to this edition of our lectures. Today we are going to be discussing about chemistry of sugars, lecture 2. In the previous lectures, we discussed about chemistry of sugars, lecture 1, whereby we introduce the concept of carbohydrates, their definition, and also the classification of the carbohydrates. And in today's lecture, we are going to be covering about the most important or the most physiologically important hexoses. We'll talk about glucose, galactose, fructose, and mannose. We are also going to talk about the roles of carbohydrate in cell membrane. So at the end of this lecture, you should be able to understand the various functions of the most important hexoses, and also you should be able to describe the role of carbohydrates in cell membrane. So let's look at this physiologically most important hexoses: glucose, galactose, fructose, and mannose. They are considered as the most important, the most physiologically important hexoses. And we can see that from the previous lecture, we understood that glucose is the most important among them because it is derived from dietary hydrolysis of carbohydrates, and also it is the monomeric unit that is used to build all other components that are derived from carbohydrates. We are going to see the various functions of this most important hexoses. There are also pentoses, which they are 5-carbon sugars. They are important in nucleic acids, and also they are also found in several coenzymes. We can say coenzymes, they are organic molecules that are required by enzymes, some certain enzymes, to function in the body. You know, in our body, there are a lot of metabolic activities taking place, and those metabolic activities are being aided or are being catalyzed by various enzymes. So some of the enzymes, they require what we call coenzymes, which are organic molecules that are needed by the enzymes to function properly. So there are also derivatives of trioses, tetroses. Trioses are 3-carbon sugars, tetroses 4-carbon, pentoses are 5-carbon, as we highlighted in the previous lectures. They also formed as metabolic intermediate in some pathways of carbohydrate metabolism, which is glycolysis and also pentose phosphate pathway. There are also carboxylic acid derivatives of glucose, which are also important. They include D-glucuronate, L-iduronate, and L-gulonate. There are also deoxy sugars that are found in nucleic acid. That is sugars, pentose sugar that are devoid of OH, those that do not have one OH, or the OH has been replaced by hydrogen, as we highlighted in the previous sugar. So you know DNA has the oxy sugar, while RNA has the sugar with OH, okay? So amino sugars are also important. We know amino sugars, they include D-glucosamine, D-galactosamine, and D-mannosamine. So if you can see, this most important physiologically active, or the most important physiologically important hexoses, you can see they are all being part of what we have discussed. You can see in D-galactosamine, the monosaccharide unit is what? is galactose. It is derived from galactose. This is from mannose. Okay, this is from glucose, and so on and so forth. So the physiologically important disaccharides includes, we have maltose, which we said maltose is a disaccharide consisting of two sugar units, two glucose units, right? Glucose and glucose form maltose. We have sucrose. Sucrose is a disaccharide that consists of two sugar units, fructose and glucose. And also we have lactose, which is a disaccharide that consists of what? Galactose and, and uh, galactose and glucose. So we also have starch and glycogen, which are storage polymer of glucose in plants and animals, respectively. So the storage form of glucose in animals is glycogen, while the storage form of glucose, or rather carbohydrates, in plants is the starch. Starch is the major source of energy in the diet. It forms an alpha glycosidic chain. It means an alpha glycosidic chain or alpha glycosidic linkage that is called glucosan or glucan. There are two main constituents of this starch. We have the amylose, which consists of about 13 to 20% of the carbohydrates, and also we have what it has non-branching helical structure. It is the amylose is non-branching, and we have the other component, which is the amylopectin, which is consists of branch chain. It is about 80 to 89, 87%, and it is composed of 24 to 30 glucose residues, and the linkage is alpha 1,4 linkage of, and also because it is branch, it has alpha 1,4 linkage in the chain, and also it has alpha 1,6 linkage at the branch point. So we are going to see structures, their structure, to properly understand. And there's what we also call glycemic index of starchy food, which is a measure of the digestibility of that starchy food based on the extent to which it rises the blood glucose concentration when compared with an equivalent amount of glucose. So this is the two forms in which starch exists. We have the amylose, which is the non-branch. It consists of alpha 1,4 glycosidic bond. It is non-branch, okay? And from this structure that you can see O, 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 right? This O, it means single glucose units. Glucose unit form together to form alpha 1,4 glycosidic bond, okay? The carbon 1 of one glucose residue makes a glycosidic linkage with carbon 4 of another glucose residue. The carbon, because this now second carbon, if this is the first carbon, let's say carbon 1, this is the first glucose residue, let's say this is carbon 1, now forms a linkage or glycosidic linkage with another glucose residue at carbon 4. So and if this glucose residue is going to form another bond, it has free carbon 1 here that it can forms a linkage or glycosidic linkage with another glucose at carbon 4. So that is linkage is what? Alpha 1,4 glycosidic bond. So take note, in amylose, it is not branch, and it is consists of what? The chain consists of alpha 1,4 linkage. So this is typical, the structure, how it looks like. Then for the amylopectin, this is the branch. Don't remember, this is the amylose component, and this is what? Amylopectin. This is 13 to 20%, and this is 80 to 87%, right? So the amylopectin is branched, okay? It has branch. In the chain, normal chain, it has alpha 1,4 glycosidic bond, just like the amylose. But along the way, it has a branch. At carbon 6, it makes a bond, alpha 1,4, alpha 1,6 glycosidic linkage, for instance. And this is, this is the carbons, the sixth carbon. And if this is the first carbon, there is a linkage between this carbon and another carbon to form the branch. So this is the branch. We have alpha 1,6 glycosidic bond as the branched. So let's look at the roles of a carbohydrate in cell membrane. We have glycogen that is, we said, is the storage polysaccharide in animals. It is highly branched structure than it is even more branched than amylopectin. You remember we said starch is the storage form of carbohydrate in plant, right? It is composed of two components. There is a linear unbranched portion that is called amylose, and there is a branch portion that is called what? Amylopectin. So glycogen is now the storage form of polysaccharide in animals. It is highly branched. The branch in glycogen is even more than that is found in the amylopectin of starch. That is what this statement means. So we said it is highly branched, and there is also muscle glycogen granules that are made of up to 60,000 glucose residues. And also there is what we call inulin, and this inulin, not insulin. You can correct it from your courseware. It's not insulin. Insulin is a hormone. Okay, it's a hormone. We are going to discuss about that in subsequent classes when we come to talk about amino acid and protein. So insulin is a hormone, okay? So while inulin is a polysaccharide of fructose, and there's what we call dextrin, which are intermediate polysaccharide that are formed as a result of hydrolysis of what? Of starch. There is also what we call cellulose. You know, cellulose is the chief constituent of plant cell wall. It is found in plant cell wall. It is insoluble and consists of glucopyranose units linked by beta-1,4 bonds to form long, straight chain straightened by the cross-linking hydrogen bonds. So if I ask you now, what is the type of linkage that is found, glycosidic bond that is found in cellulose is beta-1,4 bond, while in uh, glucose, in starch, you can have what? Alpha-1,4 linkage. In amylose and amylopectin, you have what? Both alpha-1,4 linkage and alpha-1,6 linkage, okay? So also you can have both alpha-1,4 and alpha-1,6 in glycogen, but in cellulose is beta-1,4 linkage. So mammals, they do not have the enzyme that can hydrolyze the beta-1,4 bond. So that is why cellulose cannot be digested by mammals. But it is an important source of bulk in the diet, and it is the major component that is found in the dietary fiber. So there's also what we call chitin. Chitin is a structural polysaccharide that is found in the exoskeleton of crustaceans and insects, and it's also found in the mushroom. So it is found in exoskeleton of some insect, like let's say in cockroaches, in spider, okay, in all those animals that have that have what? Exoskeleton, okay? It's usually insect, right? So chitin is found in them, flies and so on and so forth. So let's look at the structure of glycogen. This is glycogen. You remember we said it is highly branched, more than what is found in what? In amylopectin of starch. So if you look at this structure, this highly branched, you can see there is this zero, this OOO. It's just, this cycle means glucose residue linked together, right? And you can also have a branch point. The blue, the white, or let's say the gray cycle are glucose residues that are linked by what? Alpha-1,4 linkage, while the blue, rather the green cycles are glucose residues that are linked by alpha-1,6 bond. So if we open it, let's say this junction, let's see, look at this junction now. We have zoomed this junction. We can see these are the structures of glucose, alpha-1,4 linkage, and also the branch alpha-1,6 linkage, okay? So this is the typical structure of starch, of cellulose rather. Of cellulose, we said it is component of what? Plant cell, right? And look at it. If this is the leaf of a plant, and if you look at it, this is plant cell, and we now zoom into the plant cell wall. Okay, now inside the plant cell wall there is this cellulose microfibrin. So if we now zoom it inside fully, we will see that these are chains of thousands of glucose molecules. Okay, chains of thousands of glucose molecules. Now this is typically how the structure looks like, and you remember the linkage is what? Beta 1,4 linkage is beta 1,4 linkage. Okay, don't forget about that. Now, this is a typical structure of chitin, which is found in the exoskeleton of insect and crustaceans. So it has these glucose residues. And if you look at it, this is an amino sugar, glucosamine, right? And you have what N-acetyl groups there, okay? So this is typically what the structure of chitin looks like. So there is also what we call glycosaminoglycans, which are complex carbohydrates containing amino sugars and uronic acid. They may be attached to a protein molecule to form what we call proteoglycans. So glycosaminoglycans, if they are attached to protein molecule, they form what we call proteoglycan. So proteoglycan is a form of what? Glycosaminoglycan, in which the carbohydrate is attached to protein. So proteoglycans provide the ground or packaging substance of connective tissue. So now this is their importance. They are important in connective tissues. Examples include hyaluronic acid, chondroitin sulfate, and heparin. They are all examples of what? Proteoglycans. There's also what we call glycoproteins or mucoproteins, which are proteins that contain branch or unbranched polysaccharide chains. They occur in cell membranes and include serum albumin, approximately 5% of the weight of the cell membrane is carbohydrate in glycoprotein and glycolipid. So now let's look at the composition of the source of these sugars, their composition, and also the linkage that is found within them. So if this is now, for example, sucrose is a disaccharide, right? So what does it compose of? You know, we say this composed of what? Glucose and fructose. So where can we find it? I find it in cane sugar, that's the most abundant source. Can find it in sorghum and some fruits and vegetables. What about lactose? Lactose is the sugar that is found in milk. It is consists of two lactose and glucose residues. One glucose, one galactose residue. So it is found in milk. What about maltose? It is two glucose unit that is linked in alpha 1,4 linkage. Where can we find it? It is found from the enzymatic hydrolysis of starch. What about lactulose? It's composed of what? Galactose and fructose. It is found from when milk is heated. There is what we call trehalose, which is two glucose units that are linked by alpha 1,1 linkage. Remember, for maltose, it is two glucose units linked by alpha 1,4 linkage. Okay, while in trehalose, two glucose units both link in alpha 1,1 linkage. Take note of that. So what about pentosis? Pentosis, 5 carbon sugars. We have D-ribose, D-ribulose, D-arabinose, xylose, and L-xylulose. So from D-ribose, you know, it's found in what? Nucleic acid, and it's a metabolic intermediate. What about D-ribulose? It's found in what? Metabolic intermediate. On D-arabinose, it is found in plant gums. Xylose is found from plant gums and also found from some proteoglycans, while L-xylulose is found as a metabolic intermediate. So what are the hexoses? Hexoses are we talk about glucose, fructose, galactose, and mannose. Glucose is found in cane sugar, fruit juices. So what is the physiological function? It maintains metabolic fuel, and also there is what we call maltose and also lactose that are also derived from the metabolic activities that are a result of the breaking down of starchy foods that is to say they are out from dietary source, right? So we can say D-fructose also is found in fruit juices, honey, and also uh, it is found from inulin. This is not insulin. Please take note. Is inulin. This is typo error. Typographical error is inulin. That's also what we call. Now, where does it? What is the biological biochemical importance? It is readily metabolized via glucose. So fructose is metabolized via what? Glucose. What about the galactose? It is found from lactose, the sugar in milk. It is readily metabolized to glucose and also synthesized in the mammary gland for the synthesis of lactose. Mammary gland uh, of women, lactating mother. So there's what we call limanose also. It is found from plant gums and is constituent of what gluco or rather glycoproteins. So thank you very much. This brings us to the end of today's lecture. So please go through the lecture material and so watch this video. If you have not subscribed, please subscribe to this video for more content. You should endeavor to click on the notification to turn it on so that whenever we have a new video, you will be equally notified. Thank you very much. See you in the next class.